Hello students, how are you all? This is Praful. I hope you all are doing fine. Welcome to One Page Biology. So dear students, in today's video we are going to understand another very interesting and beautiful topic. The name of the topic is Chemiosmotic theory. Dear students, this particular topic is part of the 11th standard NCERT biology chapter, which is nothing but photosynthesis in higher plants. So are you all ready for this particular video? Let's begin with the video, dear students. Dear students, chemiosmotic theory was proposed by the great scientist whose name was Dr. Peter Michel. And remember, dear students, he had also received a Nobel Prize in the year 1978 for the same. So let us understand what exactly is chemiosmotic theory. Now, dear students, you had watched the video of cyclic and non-cyclic photophosphorylation. If you have not watched that video, please make sure you watch that video. Now, in that particular video, we had studied the role of cyclic and non-cyclic photophosphorylation. In cyclic and non-cyclic photophosphorylation, we saw that the electrons are actually moving from one electron carrier to the another. We also saw that the photosystem 1 and 2 comprised of chlorophyll A as well as different photosynthetic pigments. So whenever the photosystems are excited due to sunlight, they emit the electrons and these electrons are carried forward by certain electron carriers. Now dear students, talking about the non-cyclic photophosphorylation, we had seen that the final electron acceptor in case of non-cyclic photophosphorylation was NADP. So dear students, whenever the NADP accepts the electrons, it's get reduced. So during this particular process, the NADP is the electron carrier which is present on the outer side of the membrane of the thylakoid. Now because the NADP is present on the outer side, dear students, whenever the NADP gets reduced because of the electrons, it will get negatively charged. So to balance the charge, dear students, the protons are coming from the stroma into the NADP. That means, dear students, the protons are coming from the stroma which are actually reducing the NADP to NADPH2. Also, dear students, we know that during the non-cyclic photophosphorylation, the electrons were not going back to its original photosystem which was pH2. So dear students, to refill the electrons in the photosystem 2, there was splitting of the water molecule which was taking place in the presence of light. So this splitting of water molecule in the presence of light is known as photolysis. So dear students, during photolysis, the water molecule was splitting into H plus ions and the electrons as well as the oxygen was also released. So these protons which are released during the photolysis of water actually increase the concentration of protons in the lumen of the thylakoids. So dear students, because of these two following events, that is, one is the NADP is getting reduced to NADPH2 and the second event is photolysis of water. Because of these two events, the concentration of protons inside the lumen keeps on increasing compared to the concentration of protons outside, that is inside the stroma. Now dear students, because of such a difference in the protons that is inside the lumen as well as in the stroma creates the concentration difference. So if you carefully notice, dear students, you can see that the concentration of protons inside the lumen of the thylakoid is much higher than the concentration of protons in the stroma. Such a concentration difference is called as a proton gradient. So dear students, because of such a proton gradient, there is a movement of proton, that is H plus ions, from higher concentration to the lower concentration. Now this particular movement of protons, that is from higher concentration to the lower concentration, happens across a particular channel. This particular channel is made up of two units which is known as F0, F. Now F0 is a hollow part of the particular channel through which the protons are actually moving from the lumen outside, that is into the stroma. As soon as the protons move from the F0 channel, there is a conformational change in the F1 subunit. Now dear students, the F1 subunit is actually made up of a particular enzyme which is called as ATP synthase. So as soon as the protons, that is the H plus ions, move from the F0 channel, there is a shift in the F1 subunit, because of which there is a formation of energy which is in the form of ATP. Remember dear students, the F1 subunit consists of a particular enzyme which is called as ATP synthase enzyme. So dear students, this particular enzyme, that is nothing but ATP synthase, forms ATP whenever the protons move from the F0 channel. So such a movement, that is from higher concentration to lower concentration, that is from lumen into the stroma, is called as chemiosmosis, and this was particularly found by a great scientist, that is nothing but Dr. Peter Michel. So I hope dear students, you have understood this small particular concept of chemiosmosis, which is part of the chapter photosynthesis. If you have understood the topic, dear students, please like the video and also do share it with your friends. If you have any doubts or suggestions, do mention it in the comment section. Also, please don't forget to subscribe to the channel One Page Biology. See you all in the next video with some another biology related topic, dear students. Till that time, take care of yourself. Thank you so much. Bye bye.